Hello, we're glad you've joined us for this live webinar, Manipulating Trypsin Digestion Conditions to Accelerate Digestion and Improve Signal Intensity for Building a Protein-Based Mass Spectrometric Assay. I'm Alexis Krauss of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. It's brought to you by Agilent. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. Agilent provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration will provide you with the highest level of confidence in their clinical research solutions. To learn more about our sponsor, please visit www.agilent.com. Now, let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question, and click on the Send button. Questions will be answered after the presentation. To enlarge the slides window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Dr. Emma Zheng. Dr. Emma Zheng is currently the Clinical Chemistry Fellow at Cleveland Clinic. To learn more about our speaker, please click on the Speaker tab found at the top right of your screen. Dr. Zheng, you may now begin your presentation.
Thank you for the introduction. So today, my talk is on how to build a protein-based mass spectrometry assay for a clinical lab with specific focus on the approaches that one can take on manipulating trypsin digestion conditions for the development of a rapid and a multiplex protein assay. So I will first review some of the common limitations associated with immunoassays, and then the reason why we're moving to a mass spectrometry, and then I will talk about the differences between the top-down versus the bottom-up assays. I will also review the criteria for selecting signature peptide and explain the different types of digestion profiles. And then finally, the overall process of peptide selection for building a sensitive and a rapid protein-based assay. So most of the data I presented here are from the first paper listed here, which is the work I did with Dr. Marie DeMarco at St. Paul's Hospital in Vancouver, Canada. So although it has been used for many years in the clinical lab, immunoassays are not perfect. For example, interferences from autoantibodies, which has been published or described in many papers, which can cause falsely low result in a sandwich assay and a falsely high result in a competitive assay. So just taking seroglobulin as one example, autoantibody is very common. About more than 10% of people are having positive on the anti-seroglobulin antibodies. And secondly, anti-reagent antibodies, which including non-specific antibodies and anti-animal antibodies. Hook effect, which is only applicable to the sandwich immunoassays. So as the concentration of the analyte increases above a certain point, the system gets saturated and the signal begins to decline. And also the lack of specificity, protein isoforms or fragments or PTMs cannot be distinguished on the immunoassay. So people have a paper published on the PTM using immunoassays, using mass spectrometry assays that can detect different isoforms of PTH. And then lastly, the matrix effect, comparison of existing HRF1 immunoassays have found significant differences in the accuracy because the interfering of HRF1 binding proteins. So the top-down protein-based assay is analysis of the intact protein. So therefore, no protein digestion is required. It seems a very good approach, but top-down mass spec assays is not a very common practice for a clinical lab because there's only a few labs now has high-resolution mass spectrometers and has a knowledge working with intact protein analysis. So here I'm showing an example of a top-down assay which we're developing at the Cleveland Clinic for the identification of hemoglobin variants. So the sample prep is very simple. Whole blood was diluted and analyzed for the LC mass spec. So for adults, you're going to see the very abundant alpha and the beta subunits at different charge states. And after mass deconvolution, the mass of both the alpha and the beta subunit is going to show up. And if the patient has a mutation, for example, hemoglobin sickle cell, you're going to see a shift of the beta mass from the normal to the -30 delta. If the patient is hemoglobin AS, you're going to see both the normal beta subunit and also the shifted beta subunits. So the bottom-up assay, which is different than the top-down, which requires protein digestion. So the analysis is based on the signature peptides, which represent the protein of your interest. And moreover, the analyzer can be multiplex in one assay, so you can measure more than one analyze in one mass spec assay. So the typical mass spectrometer used is a triple quadruple mass spectrometer running on the targeted MRM mode. So for people that don't know well of MRM, so in the MRM, the peptide with specific mass is first selected on the first quadruple, fragmented in the second quadruple, and then reselected in the third quadruple. So what we are talking about, the one transition, is one precursor to a fragment ion pair. So as a mass back-based protein assay is gaining popular for clinical applications, clinical labs are facing development challenges. And when multiplexing of your targets, there's even more considerations. So the assay has to be using a single workflow that is suitable for all targets, a rapid workflow, meaning a reasonable assay turnaround time, a simple workflow, and also the research time and the cost. So besides the general considerations, some specific considerations would be how to select your peptides, and then which peptide used for quantification. Are you using only one peptide or using multiple peptides? Ion ratios, which one is your quantifier, which one is your qualifier? And then matrix, calibration matrix, and calibrators. Internal standards, are you using the isotopically labeled whole protein or the peptide or the new world-wind peptides? And in all of this, very importantly, how to do the sample preparation, including the protein digestion step. In a typical bottom-up assay workflow, sample has to go through several steps before entering into the mass spectrometer, like a protein enrichment. It can be immunoprecipitation, immunodepletion to remove those abundant proteins in your plasma or serum, or SPE, and then protein digestion. Peptide enrichment, like the cis-kappa assay or the SCX to fractionate your sample. And then sometimes there's a sample cleanup step. And then in all of this, the protein digestion is the rate-limiting steps. It can include denaturation, reduction, oxidation, and enzymatic cleavage. And the whole digestion process can take hours to even a day, which is make it very impractical for many clinical scenarios. So here in this table, I summarize a couple of examples of the published or existing clinical assays protein digestion time. So ApoA1 and ApoB around 20 hours. And the second example is from Dr. Hufenigel's lab at Seattle, which is included a multiple addition of trypsin. CRP HF1, both overnight, and then the infliximab assay from Mayo Clinic is also very long, requires 18-hour digestion. So if you are in a lab which doing a high volume testing, too long of a digestion time is going to lower your throughput, and also causing problem is how to schedule your tech and schedule your stuff. Another big issue is sample digestion. We're going to introduce variabilities. Variation of digestion performed at different labs is a major contributor to the overall variability of the assay. And people have also observed that discordant measurement of the peptide from the same protein used for the quantification. So, for example, the figure on the right, you're going to see, so those are the peptides from the same protein. You see a very nice correlation, but sometimes if you switch to another peptide, the correlation getting very bad. So protein digestion is a critical step to the assay performance, as the completeness, the rate, and the stability can affect the assay quality and also the assay turnaround time. So to help us better understand the key factors in the design of a sensitive and a rapid mass spec assay, we evaluated several variables that could affect the rate of peptide cleavage, digestion profiles, and also signal intensities, including the declaration potential, the DP, chelation energy, CE, denaturing condition, digestion buffer, and also trypsin grade. So these are the 5 protein markers that we used in the study. So they are CRP, RBP4, CO9, TTR, and ApoA1. So the first step of any peptide selection is basically a bioinformatics practice. There are also some web-based tools that can help you with the prediction. So in this table, I summarized some of the selection criteria which I used, taking consideration of whether it can be detected, specificity, like uniqueness, present in all isoforms, do not contain any known snaps or mutation, no miscleavage. I also try to avoid peptide with PTMs or any other modifications. And moreover, chemically stable and can be easily synthesized as internal standard for quantification. So for the 5 protein markers, we have about 2 to 9 peptides pass the selection criteria. And this table is an example of the MRM parameters. So we have 1 precursor ions for each peptide, and 3 transitions were monitored. So OTP's assays were optimized, and by doing so, we actually observed an average signal increase of over 60%. So some other groups have reported similar findings, that signal intensity was much more important, is much more improved when transitions are optimized experimentally than using those prediction equations. And to accelerate the digestion process, we used a very simple protocol in the lab. So 10 microliter of plasma or serum sample was mixed with a sample buffer at 1 to 50 volume ratio. And the reason we're using 10 microliter is for either pipetting. 1 microliter of a sample is more than enough for the detection of the 5 protein markers in the study. And then denature at 99 degrees C for 10 minutes. Cool down for 5 minutes, and then adding trypsin and incubate. And at the end, we stop the reaction by acidifying and inject the sample directly to a carbon-18 column coupled to a mass spec. So with using this protocol, we measure 5 different buffers. So they are including AMBIC, trace buffer, PBS, HIPS, and ammonia acetate. We also tested 3 different trypsin grades. Sequencing grade trypsin, which is commonly used in proteomics, TPCK trypsin, which is a pancreatic trypsin with chymotrypsin inhibitor, TPCK added, and also bovine pancreatic trypsin. We also compared with the proteomics protocol that including using SDC salt in AMBIC reduction, alkylation, and a sequencing grade trypsin for digestion. And each of the buffer system combination was digested for half an hour and also 18 hours. And finally, we have about 5 to 10 microgram of protein sample was injected to the mass spec. So there's a lot of data being collected from this buffer trypsin experiment, and which can be viewed at either the peptide, the protein, or the protein panel level. So here I'm just using plasma CRP Peptide 1 as an example. So in this figure here, the red dots are the peptide peak area at a half an hour digestion, and the blue bar are the fold change in terms of the peak area from half an hour to 18 hours. And the best buffer trypsin combination are the ones that can produce abundant signal at a half an hour and a small fold change about 1 from half an hour to 18 hours. So that means the reaction is fast, is stable, and is also non-decaying. So for example, in this figure, in the PBS sequencing grade case, the peptides produced at half an hour are very low, but there's a big increase from half an hour to 18 hours. So this is a slow reaction. So in general, the buffer studied here showed considerable differences. However, variations as a result of trypsin grade selection was far greater than the buffer systems. So bovine trypsin produced an abundant signal at a half an hour, but generated a fold change below 1 in most of the buffer tested. So that indicates a loss of peptide signal from half an hour to 18-hour digestion. And this phenomenon is most likely due to the cleavage by chymotrypsin, known to be a contaminant in the trypsin purified from pancreatic source. The genetically modified sequencing grade trypsin has a slow rate of peptide production earlier, but a big signal increase from half an hour to 18 hours. And lastly, the TPCK trypsin produced a medium intensity at an earlier time point, and the reaction is very stable and non-decaying. So given all the above results, we found that TPCK trypsin is favored as it produces a rapid and a stable signal, and also is cost-effective. If you compare the price between the TPCK trypsin with the sequencing grade, you are finding that the TPCK trypsin is actually 1,000-fold cheaper than the sequencing grade. So when searching for the best performing the buffer trypsin combination and only looking at the half an hour and 18-hour time point, there was very little variation from peptide to peptide within the same protein. So by summing the all 5 peptides from CRP together, we found that AMBIC plus TPCK trypsin is the best combination. However, for the 5 protein markers we tested, there is a significant difference from protein to protein and between plasma and serum. So what's the best combination for all 5? In plasma, we found that almost all of the buffer trypsin combinations are favored, resulting in a relative high signal at half an hour and a fold change closer to 1, as indicated by the green arrows here. But in serum, the trace buffer plus TPCK trypsin is a preferred combination. So with the consideration for the assay turnaround time, we also did a time course experiment, and monitor the digestion profiles from 5 minutes, so it's early digestion, to 24 hours. So this test was done by using the buffer trypsin system selected from the previous experiments. So peptides can have very different digestion profiles and display very different patterns. They can be slow forming, fast forming, decaying, or sometimes having these unusual profiles with signal going up and down and up and down again. And the peptides which is fast forming is the one that we are looking for, which is best for the development of a rapid assay. So just showing some examples of the profile. So here, Y is the percent change in peak area, and X is the digestion time. So in general, we observed a rapid release of peptides when using the TPCK trypsin as compared to the sequencing grade. And in this plasma CO9 plus AMBIC buffer, all of the 5 peptides of CO9 are slow forming when using the sequencing grade trypsin. But the reaction reached the maximum at a very late time point, around 18 hours. However, when switching to the TPCK trypsin, the reaction become much more faster. For example, the figure on the right, the P1 peptide plateaued at 5 minutes, and the signal is very stable for the time course we examined it. And also, peptides from the same protein can have very diverse profiles. So this finding is consistent with the poor correlation of the mass spec results with multiple peptides from the same protein were used for quantification. And using plasma TTR as another example, TPCK trypsin demonstrated a faster release of all 5 peptides compared to the sequencing grade, with a comparable maximum signal intensity if change the y-axis to the peak area instead of percent peak area change. And when switched to the PPS buffer, we found that different buffer systems can affect digestion profiles, and the peptide may perform very differently. In this CO9 case, comparing the TPCK trypsin in PBS and in AMBIC buffer, so P2 is faster forming in PBS, but it isn't in AMBIC, whereas the faster forming peptide of P1 in the AMBIC buffer showed very unusual profiles with signal going up and down again. And we not always see faster release peptides. In this case, CRP in PBS buffer, no peptides are fast forming. And how about in the serum plus tris buffer? So RBP4 is a very small protein with disulfide bonds and many PTMs. So only 2 peptides are considered for analysis. P1 is slow forming. It signals increases to 18 hours, and the P2 peaks at 20 minutes, stable to 2 hours, and then start to decay. And for serum ApoA1, we found 2 peptides are fast forming, P1 and P9. So all of the data above basically show that by selecting the best performing peptide, the overall protein digestion time can be shortened. Like if your reaction finished in 20 minutes or even 5 minutes while doing overnight digestion. And we also observed no signal increases when excess amount of trypsin was used in the system. And we found that the TPCK trypsin stock is very stable. It can be stored up to 6 months at -20 degrees C. So the next question is heat denaturation time. Can it be shortened as well? So here we tested 3 denaturation time at 0 minutes, means no denaturation, 5 minutes, and 10 minutes, and examine the profile of digestion. So taking, for example, CRP and no heat denaturation, there's not many peptides are formed before 1 hour. But if you look at the profiles of the 5 minutes compared to the 10 minutes, they are very similar. So 5 minutes is probably enough to digest CRP. So so in ApoA1, something is very interesting. We observe that there's 2 peptides, P4 and P9, are fast forming independent of heat denaturation. So if you are going to use P4 or the P9 into your analysis, the heat denaturation step can be eliminated. And finally, we are able to multiplex the 5 markers into one assay. So by selecting the best performing peptide from each of the 5 protein markers, and we and then combined into a 12 minutes LC gradient, we achieved a 20 minutes digestion by using a single workflow. And in addition, there's only less than 1 microliter of plasma sample is required, so very minimal of sample volumes. So using less sample is a big advantage for mass spectrometry assay compared with those immunoassays. And it is kind of important for a pediatric setting or for people doing research when there's not many sample is available for analysis. Consider a preterm baby in the hospital, in the ICU, how much blood he or she can provide, and also for people that are doing research using animal models, for example, mouse, how much the blood that the mouse can provide, and how much of the blood that people have in the biobank. So just a little summary of the study. The DPC optimization is important, which we yielded a greater 60% gain in signal. The buffer trypsin selection is critical. Heat denaturation is also critical, but it's only for certain proteins, but not all. AMBIC buffer plus TPCK trypsin produce a more abundant signal and a stable profiles. So by using the approaches described here, we have developed and validated a plasma ApoA1 assay, and by using one fast-forming peptide, we achieved a 20 minutes digestion time, and the measurement showed a very good correlation with nephrolometry. The assay has a lower limit of quantification at 0.01 gram per liter. And some final words about this presentation. So the approaches described here could be adopted to other protein markers. And a simple and rapid sample preparation workflow will increase the feasibility of implementation of a protein-based mass spectrometry assay in the clinical lab. And we, in the lab, Dr. DeMarco's lab at St. Paul Hospital, we actually use the same strategies applied to our development of cerebroglobulin mass spectrometry assay. At the end, I would like to thank a couple of people at the Cleveland Clinic, Dr. Wang, Dr. Ranex, and Dr. McShane, and also Dr. Maria DeMarco from St. Paul's Hospital in Vancouver, Canada, who supervised me on the work presented here.
Thank you, Dr. Zheng, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Emma will answer as many questions as time permits. So the first question is, No peptides pass the MRM selection criteria after trypsin digestion. What is the next step to take?
So trypsin is pretty common for proteomics, and but besides trypsin, there are many other proteases you can use. There's an online tool called Peptide Cutter, which you can paste your protein FASTA file, the protein amino acid sequence, and then select the different enzyme types, and it's going to show you the resulting peptide sequence. So trypsin is not the only protease. There's definitely more on the market you can buy and you can purchase and use for your study.
Now, is the TPCK trypsin commercially available?
Oh, yes. TPCK trypsin is commercially available. But the one thing I have to keep in mind is that not like the sequencing grid, it cannot prevent self-eating. So what you need to do, you need to make sure that there's no sequence similarity between the trypsin and your peptide targets.
Now, Emma, our next question, is measuring 2 or more peptides from the same protein for quantification better than using just one peptide?
I would say yes for this question. If the cost is not a problem for your lab, obviously, if you're using more peptide for quantification, you're going to need to purchase more internal standards, and the heavy labeled can be very expensive. But the good thing about using more peptide for quantification is that you can check the correlation between the results from your peptide 1 to the peptide 2. And if the patient have, for example, a SNP or mutation that he doesn't know, and then you're going to see in that case, you're going to see a very poor correlation between the two measurements. So it's pretty good way to ensure your quality and also for checking the patient's results.
And it looks like we have time for one more question. Is there any application tool that is able to help with the building and analyzing a MRM method?
Yeah, there's one which I find is pretty good, and I'm actually using. It's called Skyline. It's developed by the McCall's lab from Seattle. So it's open access that you can just download from their website. So it can help you with multiple things. It can help you with peptide selections, building your MRM. It can also help you with data analysis, which is, I know most of the mass spec vendor have their own software, but this one just can do everything in one package. So it's pretty good.
I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information that you provided at the time of registration. We would also like to thank Agilent for underwriting today's educational webcast. This webcast can be viewed on demand through August of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.